In this video, we're going to look at protein folding. We're going to see how a folded protein goes from high energy and a high entropy to low energy and low entropy. We're going to look at how we can isomerize disulfide bonds. Eventually, we're going to look at chaperones, amyloid diseases, and misfolding of proteins. And to really appreciate how a protein folds, we're going to start with Leventhal's paradox. And Leventhal's paradox goes like this. Imagine if you have a protein that is 100 residues, and each residue of that protein has 3 possible stable conformations. That means the protein has 3 to the power of 100 different combinations it can adopt, and we're going to say only one of those is the correct conformation. And let's say you could switch one of these conformations every 10 to the power of minus 13 seconds, or roughly 0.1 femtoseconds. What that means is your protein would fold on average in 3.2 times 10 to the 79 years, or much longer than the age of the universe. Now, we are living organisms, and an average lifespan of a human's 80 years, so our proteins must fold quite faster than this. So, in actuality, many proteins fold in seconds, if not milliseconds, for small proteins. And they do this by following a directed folding pathway. And the idea is that first you have secondary structure elements form, so you have your alpha helixes and beta sheets, and then you have your hydrophobic core collapse, where the hydrophobic effect goes in or is into play. All your hydrophobic residues are pushed towards the center, and you get what's called a molten globule structure, where you have correct secondary structure, but your tertiary structure isn't correct. After your molten globule stage, roughly 5 to 1,000 milliseconds later, your tertiary structure starts to form. And it does this by forming subdomains. And finally, you have rearrangement of internal side chains to get good van der Waals interactions, and you kick out any water from the core. And the idea is this is hierarchical. You have small local elements first, and then larger elements. Kind of like what's going on in this picture. You have your spaghetti, you form secondary structure, your secondary structure starts to collapse into a folded protein. That is kind of what we're talking about here. And the best way to think about how a protein folds is through what is called a folding funnel. Now, folding is very cooperative in that protein folds in parts, and that leads kind of to a chain reaction where it continues to fold. Now, the unfolded protein has very high energy, which is a bad thing. In terms of energy, you want low energy, but it has very high entropy, and that's a good thing. High entropy is good. And that makes sense for an unfolded protein. It can form a lot of different conformations, but the water has to interact with the hydrophobic residues, so that is high energy. And as it folds, a protein usually has a specific path or paths this goes down to. And so for this folding funnel, what you're seeing is that as it becomes more natured or more natural, the energy decreases and your entropy also decreases. And you can see in the folding funnel we have some hills here. These hills are called local minima. That is, a protein can reach this minima, and then you're in a good energy conformation, but it's not the best energy conformation you can be in. So to get to the best, you have to go over this hill. You have to put in some energy to break whatever favorable interactions you have. That is what an energy minima is. And so proteins can be trapped in these energy minima, and they create what are called intermediate states. And you just need some kind of random movement to get out of this intermediate state. And it's thought that proteins have evolved their primary structure over time to have the most efficient folding pathways possible, because a lot of proteins only work when they are folded. Now, if we look at protein foldings in vitro, that is outside the cell, compared to in vivo, which is inside the cell, we see that proteins actually fold slower outside the cell. And that's an interesting thought when you think about the only thing that dictates if a protein will fold properly is primary structure. So why does it matter if you're inside the cell or outside the cell? And one of the answers for that is the protein disulfide isomerase. So during folding, what could happen is that two cysteines will come into contact and form disulfide bonds. And these disulfide bonds are not present in the native state, and so they need to be eliminated. The protein that will break these disulfide bonds is disulfide isomerase. As we can see in this picture, we have a disulfide bond right here. That's not supposed to be correct. It's supposed to go 1 to 4 and 2 to 3. We have 1 to 2 and 4 to 3. So PDI will come and form a disulfide bond with the protein. That will lead the cysteine residue 2 free to attack residue 3, which will free up residue 4. Residue 4 is now free to attack residue 1, which will free PDI, and now you have the correct number of disulfide bonds. And you also have, if PDI is oxidized, it can be used to create new disulfide bonds. So here we have a reduced protein, and PDI in the peptide will form a disulfide bond, leading to attack, and now you have a native protein. But the PDI here has to be reformed. And for PDI, it's thought that it first binds to the protein via hydrophobic patches. And that makes a lot of sense. These hydrophobic patches would only be available if the protein wasn't folded properly. Otherwise, the hydrophobic patch would be buried. So if PDI sees a hydrophobic patch, that's a signal that this protein is not folded correctly yet. So it's going to bind, and through that binding, it leads to the rearrangement of disulfide bonds. So that's one reason why proteins will fold slower outside the cell than inside, because on the outside of the cell they are lacking this PDI. And that's it for this video. I will see you in the next one.